that over. I'm your host, Dr. Gillian Forrester, and you have just arrived at the second Science Saturday. Shortly, I'm going to be joined by my two guest speakers, Dr. Giulia Zanetti and Professor Adrian Shepherd from Birkbeck, University of London School of Science. But before I tell you about today's topics and I invite in our speakers, I want to tell you a little bit about what Science Saturdays are for. We've developed Science Saturdays as a series of free short talks, and they're aimed at just sharing science with the public. And we're a collaboration with the Me Human Project and the National Saturday Club. Now, if you haven't heard of the Me Human Project before, we're a public engagement project, and we're focused around sharing science and doing citizen science with the public in order to better understand our place in the natural world. The National Saturday Club is a charitable organization, and they're involved in making higher education accessible to youths aged 13 to 16 across the entire nation. So I'm here every Saturday morning this month, and I'm going to be covering a new topic with two new scientists each week. And our topics are going to range from evolution to development and from climate change to DNA. And this is so we can showcase the breadth of science and some of the pretty incredible research going on at the School of Science in Birkbeck, University of London. So you might be wondering how this is different from other science podcasts and programs. Well, our primary objective is to make science relatable and enjoyable to a general audience. And we're not only interested in the science, we also want to explore a couple of other things, like how do the scientists find their way into their current positions? So you might think about scientists needing to know exactly what they want to do when they grow up from an early age. And that's actually maybe not the true story for most of us researchers, including myself. And I do evolutionary and developmental psychology, and I work with children and chimpanzees and gorillas and orangutans. And to be honest, I didn't even know my job existed when I was a student. So we want to explore the way in which our different scientists found their way into their current roles. But we also want to explore something else, something that you probably don't normally associate with science, and that's creativity. So for me, creativity plays a huge role in the way that I do my science. But I think it's not the normal perspective. I think the normal perspective is that scientists are rather detail orientated and we've got a keenness for numbers, maybe wearing the white coats. But actually, I think there's a great deal of both art and creativity that get integrated in the way that science is created and delivered. And I want to discuss those points with our panelists today. So don't forget, you can join in the discussion as well. You can just type your questions into the chat bar and I'll be picking those up and posing those to our speakers. So we'll have time for a couple of questions after each speaker, but we'll also have time for questions at the end where both speakers will join us for a little bit of a panel discussion. So again, I just want to let you know that we're here every Saturday in May. And this is the perfect opportunity to listen in if you're just maybe having a long lie-in, or you might be going for a morning stroll, or maybe you're just having a cup of coffee with the grandparents or the kids. This should be science for everyone to enjoy. All right, so last week we brought you two talks on life in the universe and beyond. So I had my little planet here because we were talking about big things on a very large scale. And you can still check out these talks on Birkbeck's YouTube channel. Just search for Science Saturdays. But today our topic is going to take a 180 turn. Today we're going to the tiny. We're going to look at the microcosm, the tiny things inside us that make up the strange and incredible organisms that we are today. And that is why I've got my poop emoji with me today. There is a stat or a myth or truth, I'm not really sure yet, that bacteria makes up more of us than our actual human cells. And we can get our scientists to verify that or debunk it. So there's my emoji to represent today's topic. So now it's time for us to meet our speakers. And I'm going to invite in Dr. Giulia Zanetti and Professor Adrian Shepherd to our screen now. So hi, Giulia. Hi, Adrian. Can you hear us okay?
Yes, thank you.
Fantastic. Now, Giulia, you're going to be talking to us about how our cells transport information. Is that right?
Yes.
Cool. And Adrian, you're going to talk second, and you're going to tell us about antibodies and viruses.
I am.
Great. And I think this is not specific to the coronavirus, which I'm sure everybody's going to be interested in how this relates to the coronavirus. But I think this is more general.
I'm going to use flu as an example, actually.
Okay, perfect. Great. I'm sure we're going to have lots of questions for both of you. So we are ready to kick off. And at this point, I'm going to ask Giulia to share her screen with us and get her slides ready. Adrian and I are going to sit in the background, but we're here and we're listening. And I'm going to be gathering up your questions from the chat bar. So don't forget to type your questions in to the chat bar if you have any. And we'll be letting Giulia take front and center stage now. So I'm going to silence my video and my audio, but we're here and listening. And take it away, Giulia.
Thank you. My screen is working? Yeah, good. Okay, so thank you very much, Julian, and for the invitation to speak at this outreach activity. So today I'm going to tell you about public transport in our cells. But really the main focus of my talk is about proteins and how we look at proteins, how we study their structure to understand how our cells work inside the mechanisms that keep our cells working. So let's get to proteins one step at a time, and let's start from the kind of most complex form of life that we know, which is us, animals. And we can look how we are made, and we are made of organs. And then if we take each organ and we look how these are made, they're made of various tissues. And then if we look at inside tissues, the tissues are a collection of cells. So cells are the kind of unit of life of our organisms. Now, if we really want to understand how life happens, we need to understand how cells work, what happens inside our cells. So if we look inside our cells, we see that these are extremely complex assemblies. And I like to compare them to cities, which are always buzzing with activity, and they consume energy to carry out certain processes. So cells are made of a number of different components. So they are made of membranes, so that these are lipids that kind of separate cells into various compartments, and also enclose cells to separate from the outside. They're made of proteins, which is going to be the main focus of my talk today. And proteins are macromolecules. They are a chain of these molecules called amino acids, and they do almost everything inside the cell. So if you take the city comparisons, proteins are like the citizens of a city, the workers. They do most of the jobs in there. A lot of them provide structural support, and they carry out enzymatic reaction. For example, they make the energy from a sugar, they duplicate our DNA when we need to grow. They fight against pathogens, so you're going to hear from Adrian later about this. They can regulate other proteins. They can also mediate transport of other proteins, and this is what, in particular, my interest in my lab is on, so I'll focus a little bit more on this. Inside cells, we also have DNA. So you all know about DNA. Actually, DNA doesn't really do very much, apart from holding the information, the code to make the proteins that then actually do the work. And then there's also RNA, which I don't really have the time to go into, but it's a very interesting molecules, and one of the major jobs of RNA is to act as a messenger between the DNA and the proteins, and also to regulate many of the protein's activities. And then there are a number of other small chemicals, just sugar, energy molecules, etc., etc. So if we really want to understand how cells work, we need to understand how these molecules work and how they interact with each other. And I like to have this comparison. So I said a cell is like a city. So now imagine that you are an alien and you're kind of very curious and you see this planet where there's some cities buzzing with activity and you really want to understand what's going on in there. And so you can see things are moving, you can see, I don't know, smoke coming out of chimneys, etc. And if you really want to understand what's going on, you need to take a very powerful telescope and zoom in and see exactly how the people in these cities move. For example, you might take your telescope and find out that the people in the city move because they have two legs and they put them one in front of the other to move forward. And then you will see that there's also other mechanism for moving. One can walk on their legs or on wheels, for example. And so this is basically what we do with proteins. We and the other macromolecules in the cells. We want to look at them very close to understand their shape and their chemistry and to understand how they really work. So this is an example of a motor protein, which is called kinesin. And it's a protein that walks along another protein. So there's some proteins called microtubules, they form tracks, and the kinesin walks on them. So this is a movie, this is fictional, this is not data, but it's based on data. And this is the shape of the protein. And because of the chemistry of this protein, it's able to consume energy and move one leg in front of the other, exactly the same way as we walk. So we are able to understand how that particular protein works by looking at its structure. And there are many, many, many different proteins inside the cells. Each of them has different shapes and each of them does different jobs. And in the end, what we really want to understand is how these shapes relate to their function. And I just have here, there's a random one of these proteins that I've kind of looked at. This is its chemical structure, and we can zoom in into these proteins, and in the end, it all boils down to its chemistry. So these are the carbon atoms represented here. So the chemistry of the proteins and how the various atoms relate in shape to each other and to other proteins determine how these proteins actually work. So it is extremely complex. It is a wonder that we actually are able to exist at all. But if we zoom in and in and in and in, we realize that it is just chemistry that makes us alive and keep going. And this is what we structural biologists like to study. And so in order to actually see the shape of proteins, we need some very, very fancy pieces of equipment because proteins are extremely small. So you know that, so how small? Proteins have various sizes, but on average, you can put 1 million proteins one next to each other and they will make up about 1 millimeter. So they're extremely small. Another way to look at this is if you imagine to squish the whole of the UK into 1 millimeter, then your garden ants will become about the size of a protein. So they're extremely small. So we can't clearly see them with our naked eye. We need microscopes. And microscopes have various degrees of power. And the more powerful they are, the smallest the things we are able to see. So you might be familiar with light microscopes. You would have used them at school probably. Some of you might have them at home as well. And this allows to zoom in and see kind of fine details we cannot see by eye. For example, this is the eye of some fly, and these are cells that you can see the entire cells. But if we want to go and look inside cells, then we need to increase the power of our microscopes. So there are some very fancy type of light microscopes that can see inside cells. But to look at the fine details of proteins, we need to go into electron microscopes. So those are powerful enough to allow us to see details of the chemistry of the proteins. Now, this is not the only technique. A more famous technique that you probably heard of is X-ray crystallography that also allows us to look at protein structures. But what we do in my lab is electron microscopy. Right, so Oh, by the way, if you are interested in understanding how a microscope works, I would definitely recommend to check out this Lego microscope. If you Google it, there is a paper on bioarchives that has the instructions on how to build one, and that really will teach you exactly how the optics work in a microscope. This is the one that we made at home, and you can really see cells quite nicely. So that's probably a fun project that all of us can do. Okay, so back to the idea of wanting to look into proteins at very fine details. The reason why we need a very powerful microscope is that we need to look at things at very high resolution. So the idea of resolution is kind of represented by this, the word resolution that I put here. So the resolution is increasing here as we go from the R to the N. And basically it means that we can distinguish details that are closer and closer to each other. Okay, so with electron microscopy, we want to look at resolutions that are really at the atomic or molecular level. So that's the most powerful we want to go to. As I mentioned, we're talking about picometers. Right. So the electron microscope, so you can see, if to see things very, very small, we need a very, very big microscope. So this gives you an idea of the size of a microscope. This is how it looks like inside. This is a kind of a top-end microscope that we use for protein structure. And in a nutshell, it's very similar to a light microscope. So we have a source of electrons, which is like your light, that goes through your specimen. And the image is focused by lenses. Now, the difference between a light and electron microscope is that we don't have light, we have electrons, of course, and also that we can't use glass lenses because they don't do anything to electrons. We need to use lenses that are magnets. And then it all needs to work in a vacuum, otherwise the electrons are absorbed by the air. So apart from that, the basic principles are the same as for the light microscope. And we are very lucky at Birkbeck to have one of the top-end microscopes that we can use for protein structures. So this is the lady in charge of having it working on a day-to-day basis. And there's a high number of people using this microscope for their research at Birkbeck. So to look at the structures of proteins. So what my lab works on in particular is protein transport, and this is where the title of my talk comes from. And we are interested in understanding how the proteins that have their function are transported from one place in the cell to another. So here I need to tell you a little bit more about the structure of cells. So cells, as I said before, they are made also of membranes, and membranes divide, and this is in particular for eukaryotic cells, so like the cells that made our body. They are divided in compartments that are separated by membranes. Now, each compartment has a specific function. So if we go back to our city analogy, each compartment is like a different building with a particular dedicated function, like a hospital or a school or a factory. Okay, in each of these buildings, we need a very specific subset of specialized protein that would do the job. We need the doctors in the hospital, and we need the teachers and the pupils in the school, for example. And we need to make sure they don't get randomly mixed with each other. Okay, otherwise everything would just stop working, and that's what we call death, really. So if you imagine that all the school kids could be let loose at any time to roam around and go to the hospitals, you can imagine that this would cause quite a lot of problems. So we need to keep the specialized proteins in their environment, but we also need to be able to transport them from one to the other when needed. Okay, so sometimes we need doctors to go out and visit people, or we need goods coming from a factory to reach another destination. So these proteins need to be actively and selectively transported only when and where they are needed. And this is the process that we study in my lab. And it's a process that occurs via a mechanism that we call vesicular transport. Okay, so yes, so this is analogous to public transport in the city. So how does vesicular transport work? So this is just a schematic diagram of a membrane compartment inside our cell. So there's some protein inside the membrane compartment, and there's some protein outside in what we call the cytosol. There's actually some proteins that like to sit across the membrane. And what needs to happen is that this purple protein here needs to get there, okay? But one caveat is that we don't want this protein to actually ever come in contact with the cytosol. Imagine you need to go from A to B and outside is freezing cold. What you would like to do is to have direct access from your building to the underground and then direct access to the other building. This is what happens with the proteins. They don't want to be exposed to the cytosol if they are inside an organelle. And the way that this is achieved is by vesicular trafficking. So basically, what happens is that the membrane is kind of forming these bubbles, these buds, and the protein that needs to be transported is going inside these buds. And then the membrane is pinched off to form what we call a vesicle. So this is a little tiny membrane-bound compartment, which is basically a piece of the intracellular compartment that it has detached. And this travels towards the destination compartment where the membranes fuse and the purple protein then is released to its destination. So how does this actually work in details? How is the chemistry of this process? That is achieved by proteins, as all of the other functions in the cell. And the proteins that do this job of pinching out a membrane and forming these transport vesicles are called coat proteins. And this is the idea that they work by assembling with each other. So each individual coat proteins, they bind to each other to form these scaffolds that form these kind of cages, and they pull out the membrane to form these coated vesicles. And at the same time, they're also able to include the proteins that need to be transported inside these budding vesicles. So this is the proteins that we study in my lab, coat proteins. And we want to understand what their shape is and what their chemistry is and how they interact with each other to form these scaffolds, how these processes are regulated, how is the energy used to bend the membranes to form these kind of structures, which here are spheres, but actually there's many different shapes and sizes that can be formed. So the coat proteins have a number of properties. One of them is that they need to do something that is quite hard, because membranes, if they can help it, they'd rather be flat. So one of the jobs of these coat proteins is to pull the membrane to form these spherical vesicles, which require a lot of energy. They also need to be selective, because they don't just form vesicles to transport everything. That would be equivalent to random mixing. So they need to really select who's getting into the vesicle and who's staying out of the vesicle. They need to know where to go. So if we take a certain compartment and we form a vesicle, that vesicle can't just go anywhere in the cells. It needs to know its destination. And this is achieved again by the coat protein complexes. And also, as I mentioned already, they require energy. So understanding the structure, the chemistry of these proteins will really help us understand how all of these properties are achieved and really understand how the membrane deformation happens. And so in my lab in particular, we study one pathway, which is going from one organelle, which is called the endoplasmic reticulum, or ER, to another organelle, which is called the Golgi apparatus. And so this is a pathway that is mediated by a particular set of coat proteins that are called the COP2 coat. And what we do in the lab is we basically reproduce the process of membrane deformation and vesicular budding in a test tube. So we take all of the proteins belonging to the COP2 coat. We take artificial membranes and then we mix them in a test tube so they form these vesicles, and then we use the electron microscope to look at the shape of the proteins around these vesicles. Now, this is the only piece of data that I'm going to show you, and this is a typical image that we get from the microscope, and then we need to spend a long time, months and months, doing image processing in the computer to retrieve the signal from these images and remove the noise so we can see the fine structure at the molecular level of these proteins that are assembling and forming these vesicles. Okay, so this is just a final movie that I want to show you. It's taken from YouTube. This is not a movie that we've made. And it's a little bit also the actual chemical interactions that are represented in these movies. Some of them are correct. Some of them have been corrected later on by further research, but it will give you a pretty good idea of how the COP2 coat actually works to perform its job. So here we are inside the endoplasmic reticulum, so the starting organelle. So we are inside the lumen, and this protein want to get out inside these vesicles to reach the Golgi apparatus, these yellow and purple proteins. So now here we are outside, and these are all components of the COP2 coat. Please ignore the names of the proteins. So we have a first protein that docks onto the membrane of the endoplasmic reticulum. This is the energy molecule that is bound. And when the energy molecule is bound, this yellow protein recruits by chemical interaction these other proteins. Now, this represents the true structure of these proteins. So we know that this is the actual chemistry of how they interact with each other. And then there's recruitment of another set of proteins. You see this kind of very long bar-shaped proteins that are recruited. And these bar-shaped proteins, they like to bind to each other to form polyhedral cages. Now, because they have this tendency to form polyhedral cages, eventually they pull out the membrane because they are also stuck to the membrane via the interaction with the yellow proteins. They pull out the membrane, and that's now the membrane full of those proteins that wanted to go out and be transported. Okay, so eventually then this vesicle loses its coat and is transported to the final destination, possibly along these tracks through these walking proteins, as I showed you at the beginning. Okay, and this is another way that you can look at the proteins. This is something that we've done at home with plasticine and stop motion animation. You can sort of try and reproduce how these processes are working, and this really helps understand, each step of the process. And there are a number of videos on YouTube with many different projects with plasticine and stop motion to understand various biological processes, which I strongly recommend to look up. Okay, my one last slide is just to mention that doing protein structure is also one of the fundamental techniques at the basis of drug design, because if we know the structure and the chemistry of the proteins, then we can design molecules that bind to these proteins. And very often diseases are caused by proteins that are dysregulated and don't work properly. So we can design drugs that will inhibit these proteins and kind of solve the disease. So that's just kind of one important aspect that I wanted to mention, even though I hope I could kind of transmit the message that we want to do structural biology, also because we're curious about understanding how these proteins actually work and their molecular mechanisms. And that's it. So just final slide to say that research is never done by individuals. It's always done by groups of people working all together. And this is like the people who are working with me. And I just kind of this is just the places where the people mentioned in the slides are coming from. So science is an extremely international and fun environment to work in. And yes, that's all from me.
Thank you so much, Giulia. God, that was so interesting. I wish I had had a biology teacher like you when I was in school with the analogies. I think it makes it so much more understandable and relatable to be able to see how these things work based on structures or functions that we already know about. So like transport systems. So I thought that was beautifully done. So thank you so much for that. I never knew proteins walked like that on tracks. That's pretty cool. And the fact that we have the technologies to see and then understand and replicate these in video form is pretty amazing. So you've told us that one of the applications of understanding this is that we can manufacture drugs that might help us for people who have different kinds of conditions. Is that the main reason that we would study this kind of transport in ourselves?
No, not for me. I mean, for some people it may be. For me personally, is to understand how things work. But obviously, it's the reason why research councils put so much money into it as well. And, there's a big focus on structural biology from pharmaceutical companies as well, is because of drug design and this huge effort in understanding, for example, the structure of the coronavirus spike proteins amongst, just to say something that is topical at the moment, to understand how to inhibit it and stop it from binding to our cells, for example.
But your main motivation, like mine, is to work out why and how we became the humans that we are today.
Yeah.
So like, what is it that makes us up and how does it function?
Yeah.
Which I think is just, yeah, you're in it for the pure research aspect of understanding who we are and how we came to be. We've got a question from one of our viewers, Chris Daniels, who's asked, how do proteins know what to do? Is it a simple program or do they think in any way? And by joining together, do they think more?
Okay, well, that thinking is probably not, I don't know, this is a difficult question, but essentially it's written in their chemistry. So proteins are chains of amino acids, which are like 20 different molecules. And depending on how you put them, in which order you put them, proteins assemble and kind of fold to make different shapes and have different chemistries. And that's what we are really trying to understand, is how the chemistry of each of these proteins makes them do what they do. Okay, so for example, you might have two proteins that to work, they need to interact with each other. And when we look at them, we find that one is positively charged, the other one is negatively charged. So they tend to stick to each other. And that's how they actually know what to do, is because the chemistry, tells them what to do. So they don't think, they just do what they need to do, because that's how they're made. And what tells them what their shape should be is the sequence of their amino acids. So in which order the amino acids are there, which is written in our DNA. So the DNA holds the code, the sequence that will eventually tell how the protein should be made, and therefore what structure and chemistry they will have.
These are pre-programmed things that are going to happen based on the pre-programming, and they're not going to do something different, really. They're not going to think, oh, I'd rather be going that way or doing this.
Well,
no,
they're not, unless they're different to what they are supposed to be. For example, if there is a mutation on our DNA that will cause a change in an amino acid in our protein, that protein might not work very well. And very often this causes disease, for example.
So those are exactly the kinds of things you'd want to understand. And that's what the drug companies want to understand, so they can manufacture different medications to inhibit those things, those mutations. Oh, so cool. So I've got so many more questions, but I think we're gonna stop there for a second. I am gonna want to ask you how you got into your field and what was kind of the trajectory from school all the way up. But I'm going to hold that to our panel because I want to move on now to Adrian's talk about antibodies versus viruses, a war in vain. And I'm going to silence my video and audio again and take it away, Adrian.
Okay. Let me just see if I can get a pointer up. Maybe not. I can do without it. Can you see a pointer there? No. Okay. So actually, I want to start, since you just raised that point. My own background is really not very scientific, so I'm going to start talking about how I got into science and preempt what's already been mentioned. So actually, I started off doing biology, chemistry, and history were my first choice of A-levels. And somebody asked me, what's that good for? I mean, I'd chosen them because that's what I enjoyed doing when I was earlier on in school. And it turns out they were good for archaeology. So I actually went on a dig, and that was the end of my career as an archaeologist, was one dig. And at that point, I changed from chemistry to English right in midstream. I was all over the place, really. And that led on to, I applied to university, and I didn't get my first choice. I ended up on studying history of art, which seems very random now. And I've studied in the building to the top right there, which is at the University of East Anglia. It's actually houses an art gallery, and it was actually really enjoyed the course. I studied things like the painting at the bottom. That's a detail of a painting that's out in Venice, which is very nice. I also studied pub architecture. And after that, well, I really didn't know what I wanted to do. I knew I didn't want to work in a museum. And after a couple of years trying out various things, I became what's known as a welfare rights officer in North London, funded by Haringey Council. I didn't spend a lot of time dressing up, but here I am protesting. I'm dressed as a revolting peasant there, protesting against something called the poll tax. I'm not going to talk about that. So a completely different direction from history of art. And then I got into computer science. And I think really before that, before this step, I thought of computers as being kind of glorified calculators. But a couple of things happened, one of which was reading a book, the book that I show here, which is making connections between science and the arts. So Gödel is a mathematician, Escher an artist, and Bach the composer. And it opened up my mind to things like artificial intelligence and various other things. So on the strength of that, I applied to do a master's in computer science at UCL. I did a project with Dr. Denise Gorse, who's an expert in artificial neural networks. And I think because I could write well, probably the arts background came in handy. She offered me a PhD position in what was then called neural computing, and is nowadays called machine learning. So that was my next stage. And at that point, I got offered various projects. One was to do with missile guidance systems. I wasn't going to do that. And another was called the virtual catwalk. I wasn't interested in that either. So I went on to study what's called bioinformatics. So this is computing applied to biology. And this is doing research in other people's groups with two excellent, very well-known scientists in the field, Janet Thornton and Christine Orengo, who taught me a huge amount, including how to run a group, I guess. So I did that for several years. And then eventually I got offered a lectureship at Birkbeck, and that's where I've stayed ever since. And actually the area that I now work in, immunology, I didn't really get into that until probably the last 12 years. So quite a late developer. This is not the standard route to becoming a professor. So I'm going to talk about the immune system. I'm going to talk about viruses and antibodies, but there's several things I want to cover. The first of which is partly because we see a lot of viruses on the screens these days, but very often the scale is wrong. So I want to give a sense of the scale. And I'm starting off here with a human cell. And human cells vary hugely in size. So this is just a fairly typical size, I guess. And I've drawn it round. They can be various shapes. And actually, I've colored it like the sun, partly because I think there's an analogy between the size of the Earth to the sun and the size of a virus to the cell. And one of the things I want to do, so this is, measured in micrometers, but I want to give a sense of how big that is in a way that perhaps we can relate to. So I've chosen the thickness of a sheet of paper, and a single sheet of paper might be about 2 human cells. They do vary a lot in size though. And now I'm going to zoom in because we need to change the scale if we're going to understand the size of a virus. And I've chosen flu virus because we've done quite a bit of work on that, and it's a respiratory virus like COVID-19. And you can see it's very small by comparison. The measurement here is in nanometers, and on the thickness of the sheet of paper analogy, then we're talking about maybe 1,000 viruses. So that's the virus, but I need to zoom in once more. And this is a model of the virus. And you can see on the surface are these protrusions. These are proteins. The virus is made from proteins. It's our own cells that do the manufacturing of those proteins. And the blue one here is called hemagglutinin. It's the critical one that the virus needs to be able to engage with the cell and enter the cell, essentially. I'll come on to that a little bit in a moment. So that's the virus. And by comparison, here's an antibody. So an antibody is another protein, and it's a distinctive Y-shaped molecule. And you can see it's pretty small, even in comparison to the virus. So the question is, how does an antibody combat, or multiple antibodies combat viruses? What do they actually do? Oh, just to say, of course, we've got the issue about size again, and I did this calculation. I don't know how accurate it is, frankly, but it's an approximation. We're talking about thousands of antibodies stacked up to meet the thickness of a single sheet of paper. So to understand what the antibody does, we have to understand how the virus gets into a cell. And this surface molecule, this surface protein called hemagglutinin, is bound to the membrane of the virus, which is the membranes here in the grey block. So that bit's just a schematic, but the top part is taken from a structure that was solved by X-ray crystallography. So we do know a lot about the detail of this protein. And up near the top is a specific site that can bind to what's called a receptor on the surface of a cell. We have lots of receptors on the surface of our cells, and some of them are part of the transport system that Giulia's talked about. So they transport things from outside the cell into the cell. And the virus down the bottom there is a kind of hitchhiker in a way. It binds to a receptor and then gets drawn into the cell. So I'm not going to talk about that bit, but the crucial thing then, at least one way, and perhaps the most important way that an antibody can stop the virus getting into the cell, is to block these key locations on hemagglutinin. And here I'm just showing a single antibody that happens to bind to 2 of these molecules, these hemagglutinin molecules. Actually, there's several locations they can bind to on this molecule, and they can do the blocking in various ways. So this raises some interesting and important questions. how come we have antibodies that can do this? Because they work against, we have antibodies that work against flu, that work against coronavirus, and any number of other microbes. And in the case of coronavirus, this is something that's new. It's not as if flu's been around a long time. So, we could have evolved to combat flu, but that's not true of coronavirus. So how come we have effective antibodies? One possibility is that each antibody can bind to lots of different things, but that's very dangerous, because if we have antibodies that bind to our own proteins, it could mess with our normal processes, and this is called autoimmune disease. And a certain amount of that happens, but that's not the key to this problem. I mean, fundamentally what happens is we have an incredibly diverse range of antibodies that can bind to different things. So it's binding to different bits of protein surfaces, essentially. And just one other thing that I also want to mention is, how many antibodies do we need? How many viruses are there? So from that point of view, an infection can start with a single virus. I mean, very often it will be more than that. And, if it's a single virus, maybe we can stop it before it gets started. But within a single cell, the number of viruses that can be produced is huge. I mean, potentially 100,000 viruses. And if somebody gets a runaway infection, then the total number of viruses has been estimated to be 1 trillion, which is a huge number. Now, I was mentioning earlier about the thickness of a sheet of paper. So I want to do an analogy now on the numbers, population numbers of viruses. So suppose we count them at the rate of 1 per second. How long would it take? Well, 100,000 viruses would take 28 hours. 1 trillion would take nearly 32,000 years. So that's a very, very big number. So how come we've got all these different antibodies? Well, the starting point is genes. So this is DNA. And the numbers are right here, or at least most of us have about 40 of these V genes. These labels V, D, and J are correct as well. Actually, these genes are arranged sequentially within chromosomes rather than stacked up here. That's just to illustrate the point. But to build an antibody, our bodies take samples from each of these piles. It's not entirely random, but you end up with a lot of possible combinations. And then on top of that, there are various other random processes that introduce additional changes to the sequence. So these are these little red vertical bars that you can see. And actually there are 2 sequences that combine together to form these kind of Y-shaped arms of the antibody. And if you add up all of this, well, a recent estimate is a ridiculously huge number of different antibodies, 3 quadrillion. That's much higher than earlier estimates. It's certainly going to be a high number. If it's 3 quadrillion and you are counting them 1 second at a time, that's 100 million years. And going backwards in time, that takes you before T-Rex roamed the Earth. Now, the final point I really want to make here is that these differences matter, and the focus for these differences and the variability within these antibodies is within the bits that are in direct contact with, in this case, hemagglutinin. So these are the letters, these are the amino acids that Giulia was talking about earlier. So the differences change this alphabet for a particular antibody, and it will bind in a different way. And that's where we get this huge population of antibodies that can bind to lots of different things. So now I just want to talk about what I do as a computational biologist. And Sometimes we work with structures that people like Giulia have solved, but actually most commonly within my group, we work with sequences and populations, in this case, populations of antibodies. And this is a previous paper, not by us, trying to find ways of visualizing what happens. So we're interested in the dynamics of the antibody response to a viral infection. And what you can see here is the red vertical line is 1 hour before vaccination. So this is the flu vaccine. And blood samples are taken at various points. And the antibody sequences are worked out. And from that, you can get some sense of what's going on. And this is called a stream graph. And what it's really showing is that there's a peak response around day 7. And to the vaccine in this case, and they're colored yellow in this particular graph, and that's showing that they're kind of different from the antibodies that were there before. This is kind of a typical response, so after about 7 days to the vaccine. But the important thing is once you've been vaccinated, you have the cells that produce antibodies, called B cells, are there waiting in case you get infected by flu itself, and you can have a much quicker and more profound response. Now, what we were interested in is this same data. So within my group, I've got a current student, Colin Stratton, and what we're currently working on, one of the things we're working on is something that tells us more information about that same data. So what's the relationships between the antibodies that we're sequencing? And in this particular image, this is kind of before vaccination. There are a few clusters, the hotspots here, where there's quite a lot of antibodies that are. So what I talked about earlier was the diversity of different unique antibodies, but we can have multiple copies of particular antibodies. And I don't know what these hotspots are here. Perhaps there's a prior infection. This is the kind of thing that we would like to explore. But 7 days after, so this is the second sample, you can see there's areas where there are a lot of expansion. And it's not just one location. And what we've had to do to work this out is we can calculate the difference between a pair of antibodies. And the real challenge here is then to project that into 2 dimensions to get some kind of meaningful visual representation of what's going on. So that's one of the things we're looking at the moment, and we want to compare repertoires. This is what we call them, a repertoire of antibodies. We want to make comparisons between different individuals to see if everybody's response is similar, or are there individuals who have hotspots in different areas and so on. And this would be interactive, so you could draw a circle around a particular group here and then get some information about them. So visual exploration of the data is often very, very important. And then the final slide. So I've talked about the antibodies, but actually the virus can fight back against antibodies, and it does this by mutating. And we know this, this is getting mentioned quite a lot in the context of coronavirus. Variants are in the news. And what I'm doing in this research, so this is research we did a few years ago looking at flu. This is a particular type of flu. This is the Hong Kong flu. So there's a pandemic in 1968. And there are 3 plots here for different locations on the virus, on hemagglutinin. And the purple arrow on the right shows you where they are. They're all close to each other. And what these graphs are showing is that at a particular point where the red arrows are, so this is around 2003, all 3 mutated at more or less the same time. So the entire viral population adopted a new amino acid at each of these positions. And this becomes a way of, from viral sequence data, to predict that it's escaping from antibodies in the human population, and this is the sort of thing that would indicate that you need to change the vaccine. And that was done by my then PhD student, Kevin Pentia. So that's it for me, and I look forward to questions.
Thank you again, Adrian, for another wonderful talk. And yes, please put any questions that you might have in our chat bar, and we can ask those to Adrian. But I've just got one or two for you before we get to those. Just out of curiosity, obviously, you've told us loads about how antibodies and viruses work together, but is there a lot of individual differences in the amount of antibodies people have? Different people maybe of different ages or across the world, or maybe newborn babies? How do these things vary?
They vary a lot. So as you get older, the response becomes slower and less diverse. If you have a pair of identical twins, as they grow older, their responses diverge, because part of your response is determined by your prehistory of infection. Also, we are genetically rather different in terms of the genes that encode for antibodies. So it's one part of our genomes that is quite different. Quite a few of the genes that encode for different parts of the immune system are the areas where we're most different from each other. So the set of antibodies that we can produce will tend to vary between individuals to some extent, at least. So does that answer your question?
Yeah. So newborn babies who don't have a history of infection, what about them?
Well, they have some kind. It's not something I know particularly about, but certain things can cross over the placenta, so they have some exposure, and I think they inherit something from maternal antibodies as well, and protection from that. So yeah, it's definitely a very, very interesting question. Yeah, all of these things are interesting. And why, for example, do children have a very different response to coronavirus, for example? And a lot of research going on in these areas.
A lot of things we don't know yet. So I've got another question, and I don't know if there's been research in this area how this works. Maybe you can explain it to us. But when you have something new like coronavirus come along, and we already have these known antibodies that are maybe genetically predetermined, do we create new ones, or do old ones maybe bind together and work together in ways that help combat new viruses?
So we have what I was describing, the kind of combinatorial thing, lots of different genes combining, means that we each have an incredibly diverse naive repertoire. So that's the background, if you like. And something new comes along, some of them will be able to bind to it, but they bind rather weakly. And there's a further process that I didn't go into where the antibodies evolve, essentially. They compete to see which can bind more strongly to the virus, and they get better. So there's it's called affinity maturation is the technical term for that.
And that's going to be individually based within that individual.
Yes. And if the virus is itself evolving, so a long-term, a chronic viral infection like HIV, for example, the two things are going on at the same time. So our antibodies are evolving. This is a kind of sometimes called the evolution within us.
Okay.
And the virus is changing as well. So the two are going on in a very dynamic way.
Cool. Fascinating. Okay, I know you didn't talk about bacteria, but it's part of the microcosm as well. And I just want to know if you can verify or debunk that statement out there that we are more bacteria than we are actually human.
I guess it depends what you mean by more. What I can certainly say is that the bacteria in our gut is one of the things that educates our immune systems. So we are patrolling this boundary between self and non-self, and from the immune system perspective, some of the good bacteria are self. They're part of us as far as the immune system is concerned. But yeah, I think it depends how you ask the question as to, I think in terms of genetic diversity, it's probably true.
Cool. And maybe numbers as well, obviously, because we know that they're so much smaller than
yeah. I mean, the population size is phenomenal.
Yeah. Cool. Thank you. Okay, I'm going to invite Giulia back in. And I said at the beginning, I was interested in not just sharing the science from the perspective of what is the science itself, but also having a think about the multidisciplinary aspect of the science and the research. And is it all just super geeky, nerdy, in the lab with your white coat on doing experiments with microscopes thing, or is there any creativity that's involved in the science that you do? So I put that to both of you. Who wants to go first?
Well, since I raised it, maybe I'll go first. So the reason I gave up chemistry actually was because I found it boring at A level. And the reason I found it boring was because it seemed to be entirely about reproducing other people's results. Whereas doing science, doing research like we do, you're always, so you're having to make judgments. You're having to use your imagination to come up with explanations and to design solutions to, in my case, it might be writing programs, understanding what we can do, the right way to visualize data, which is one of the points I made. So there's this feeling for me that the school science as I did it anyway did not represent what the excitement of doing science in a university setting.
Yeah, I
I
agree with Adrian. I mean, it's about being able to ask questions that nobody has asked before. So you're kind of going into uncharted territory, and you have all the tools at your disposal to kind of design experiments to answer your questions, and that's a lot of fun. It's not boring. It's not grey. And the other thing that obviously Adrian has a background in arts. I don't, but I find it quite amazing how you can actually make parallels and then use creativity or arts or pictures, Play-Doh or whatever to actually relieve what happens in ourselves in this case, but with science in general, and relate to people who might not have the same background as you. And I think that's very, very important.
Yeah, I agree. And I think that's where I find the creativity also comes into a lot of the work that I do. Is it's great that I can do the work and I can share that work with the scientific community, but there's a responsibility that we have as researchers to make this understandable and relatable to the wider public as well. And I think sometimes the best way to do that, just like you did, Giulia, is with analogies and illustrations. And to be honest, I feel like that often helps me as well in understanding the relationships better as well. And sometimes that drives me to ask a question that I had never thought about before. So I find that the whole cycle of public engagement and research, they actually feed mutually into each other to create better science. And I love that aspect of it. So we've got some questions rolling in. So I've got another one for Adrian here from Chris Gangles asking, can viruses mutate forever? For example, if we take COVID, do we have to keep vaccinating annually forever to deal with mutations, or is there an end point?
Well, there may be an end point in one sense, which is that it becomes less of a problem. So it's not necessarily in the virus's interest to be associated with severe disease. And so one quite likely scenario is that it will evolve into something that is more like a cold. Indeed, some cold viruses are indeed coronaviruses. But the other thing is that it doesn't evolve as rapidly as flu does. The thing with flu is that it's out there in birds and in pigs, so we can't really get rid of it in that sense. And when it jumps from another species, that's when the biggest risk is. So that's the phase we're in currently with coronavirus.
So am I right, Adrian, in saying that like the virus's goal is to keep reproducing and passing on from host to host, so it doesn't really want to kill its host? So severe disease actually isn't its goal, because that would mean that it dies with the host. And so like as it mutates, it's going to get less severe so it can pass to more people and keep spreading.
Indeed. But on the other hand, I mean, horribly, some viruses do rather well by inducing severe symptoms that make people cough and shed out lots of virus, after which the host dies, but at that point it's moved on to other people. So it you know, it can get quite a long way on being rather nasty.
And I suppose if it's jumping species, then we just have no idea how the symptoms are going to manifest when it moves into that new species. So that's incredibly scary.
Yeah, prediction of symptoms is very difficult. I mean, there's a lot of surveillance goes on to detect when a virus looks like it has potential. So it comes down to these receptors on the cells. Is it compatible with our receptors? And actually, there was a paper in 2015 highlighting bat coronaviruses, saying it looks like, you know, here's one that could jump to humans. So it's kind of down to us, I think, to monitor these things to some extent.
Yeah, indeed. Giulia, we've got a question for you from Alessandro Costa asking, sorry, the question is from Martino Costa, has a question for Giulia. Where do the cells get the amino acids to make the proteins?
Right. So many of them, they come from our diet. So we need to get them through our food. And some of them, the cells can build themselves. So about half of them, the cell is able to build from more basic molecules that need to come from our diet themselves. And then the proteins can take these basic molecules and make the amino acids. Some others, we can't make them and we need to get them straight from our food. So it's very important to make sure that we eat a well-balanced diet with that will provide all the amino acids we need.
Fantastic, cool. So our 5 a day are quite important for that?
Yeah, and for all of the other molecules that we need, of course.
Okay, excellent. That's a new pleading from parents saying, you won't make your amino acids if you don't eat your 5 a day. Get those veggies down you. Yes, from our good fibers and things as well, doesn't it?
Yeah, and kind of meat and eggs and cheese, but alternative also, you know, pulses, and there's plenty of information online on where to get the amino acids. I'm not a dietologist.
And Giulia, we touched on it earlier, asking about routes through. So you've both been doing science for quite a number of years now, but we all travel into our science research areas in very, very different fashions, taking different paths along the way. Some of us knowing what we want to do right from the beginning, others of us only finding out that our jobs even exist much later in life and having had different experiences prior to that. And I always say this to my students, it's just as important to try things out to figure out that you don't like them as it is to figure them, try them out to see if you do like them, because you might be passionate as a child about wanting to be, say, for example, I think I wanted to be a vet for many years as a small child. But then I think when I got to take some sample classes into what veterinary medicine might be like, I thought, oh gosh, no, I feel too emotional about this and it might not be the right thing for me. It was so good for me to have that experience to know maybe that didn't suit me, but I could pursue different areas of science that still worked with animals. And that's what I do today. I still work with animals. I work with great apes. But I'm not I'm not doing it in a medical fashion. So it was really important for me to figure that out and try lots of things, so I knew which pathway I wanted to take. And that took a long time. It wasn't a straight line, but it's a very worthwhile journey to find out what's right for you. So can you tell us a little bit about what your journey was like?
Sure. I mean, my journey was pretty standard in a way, in the sense that I studied sciences at university and then went on to do research. But I didn't study biology at university because I wanted to be a researcher. I don't think I had any idea of what a researcher would do. I mean, this kind of, for me, it was not like a job. I studied that because I was interested in it, and I thought it was fun. And I think that's the most important thing. And after I finished my degree, I had the opportunity to do a master's, a research project in Australia. And for me, the thing about traveling and spending a year in Australia, I was like, yeah, I'm going to take it. And not because I wanted to be a researcher, but because I thought it would be a great experience. And I fell in love with the research and with the environment. And as I mentioned, it's extremely international, and you're always talking to people who are curious and active, and it's just a lot of fun. So I just decided that was what I was going to try and do for my career. So I've always been interested in science, but not particularly in this kind of niche of doing structural biology, for instance. It just evolved.
It happened. No, and those are great points too. I think that when you're in school and you're doing your A levels, most of the stuff that you're learning about is applied, right? So it's like, oh, are you going to be a physicist, or are you going to be a chemist? Are you going to be a biologist? But I think we don't really learn at those stages of our lives that actually there are jobs being researchers in those areas, and you can go on to be full-time researchers and just study the things that you really like, do experiments in those areas, write them up for publication in scientific journals, be associated with laboratories or universities, do some teaching, do some public engagement. And alongside that often comes the opportunity to present your work and your research at conferences around the world, which will involve travel. You might also take lab placements at different laboratories in different parts of the world. So meeting new people, looking at new approaches, new areas of research. So there's so much opportunity, but I just think for me, one of the purposes of the Science Saturdays is to let people know that you don't have to be pigeonholed as a particular kind of scientist early on, and that there are the jobs available are so much broader than one might think when you're going to school. So just kind of bear in mind that if you find something you're passionate about, there will be a job in that area doing research into that thing.
Absolutely, I agree. I mean, I wasn't doing this type of science from the beginning. And I think, there's even some funding schemes that encourage people to change scientific field, because in the end, communication between people with different sets of skills and background is what really makes progress happen. Because if people are just kind of stuck in their own little world and talking to people who know the same things, then there's little chance to progress. And it's when we meet scientists with totally different backgrounds, or even people that are non-scientists, that we get new ideas and kind of we really can make leap forwards. So
yeah, and those kind of barriers, the divisions between different subjects that we get at school don't apply either. I mean, to a huge extent. And I think there should be more scope for, people should bring in soil samples from their garden, and you do chemistry and biology and all kinds of stuff on that, and try and work out what's in it, and because that's what certainly I feel that I'm doing: computing, maths, some statistics, biology, chemistry, all of these things together in my research, really. And I'm not necessarily an expert in very many of those, but you interact with people who are.
Right, and that's where the collaborations come in.
Yeah.
So breaking down the barriers doesn't necessarily mean that you have to become an expert in all of the areas. It means that you work with people collaboratively, and each of you have your specializations, but you can pool all your knowledge and your skills to create novel approaches to studying who we are and and how we became the really interesting primates that we are today, and what is our place in the natural world and how we fit into this planet and its ecosystems. So I find that fascinating too. I want to thank Giulia and Adrian so much for sharing their talks with us today and their really interesting subject areas. This video will become static on Birkbeck's YouTube channel, so you can just search for Science Saturday, the Microcosm, and you can watch this anytime, share it with your family, you can play it in your schools. We hope you will enjoy it. We hope you did enjoy it. We thank you for the questions that you've posed as well. And you can also check out Birkbeck's Twitter feed and the Me Human Twitter feed and the National Saturday Twitter feed, who are all our partners today and sponsoring this event. So just to let you know, we've got 3 more weeks of Science Saturdays coming. That's the 15th of May, the 22nd of May, and the 29th of May. That's every Saturday morning for the rest of May, starting at 10:30. You'll be here with me and 2 new speakers every week. So next week we have Dr. Emma Mayburn and Dr. Catherine Thompson, and our topic is nature and nurture. So we're going to be understanding about DNA and how everybody's DNA makes them who they are. But we're also going to hear about how the environment can change how you develop, thinking about how pollutants impact our breathing system, our respiratory system, how it impacts our lungs. On the 22nd of May, we've got a topic called Mind the Mind, and we have Dr. Simon Green and Dr. Ute Lersch, both talking to us about more the psychology of the mind. Simon Green's going to tell us about the biology and the culture of psychiatric disorders, while Ute is going to tell us about how resilience works and making resilience work for you. And finally, in our final week, where we've got a theme called Becoming Human, I'm going to talk to you about the 2 sides of your brain and how they're very much the same, but they're also very different, and how evolution has caused the 2 sides of our brain to have slightly different dominant functions. And my colleague, Dr. Natasha Kirkham, is going to tell us about how babies manage to develop in very noisy environments. So the world can feel a little bit messy, yet they can still develop typically and gain all these incredible human skills that we have today. So that's every Saturday morning at 10:30. Hope to see you next week. Thank you again to Giulia and Adrian. Bye for now.